Hey there, everybody. Welcome into our webinar. We are so excited to have you. Today, we'll be going over the dirt on soil science. As folks are kind of coming into the Zoom room, settle in, get ready for our webinar. I would like to ask you to navigate to the bottom of your Zoom window and to click on that little, it looks like a little chat bubble, and let me know. Let's see, what's a good question? What is the first thing you plan on doing when you get to summer break? So let me know in the chat box what your summer plans post testing, post finals, post grading looks like if you want. Now, make sure when you hit send that your chat is set to everyone. That's going to help us when we are going through the webinar. People are more than welcome to ask questions throughout the webinar, and we ask that everyone sets their chat to everyone so that people know what was asked and what Colleen's answering. So it looks like we've got some answers rolling in. We've got folks relaxing, spending time outside in the garden. Sounds like we have a good webinar for you. Oh, awesome. We have family time coming up, teaching. Oh, some family. Yeah, it looks like everyone has a really good time coming up this summer. That is so awesome to hear. All right, my friends, we are 2 minutes after the hour, so to make the most of our time today, I'm going to go ahead and jump into some housekeeping items. So first and foremost, you will receive a recording of this webinar. It'll be both on our YouTube and on our website. It usually takes us about 3 to 5 business days to get that up. You'll also receive an email with that. So keep an eye on your inbox. It is coming to you, I promise. And last but not least, I did also just want to note that we will be answering your questions. Please feel free to put them in the chat bubble just like you are right now. If we happen to not get to your question, please do reach out to us at biology@vernier.com. And that's it for me. I'm going to drop a link to our resources folder as soon as I'm done talking. The presentation as well as some resources are in there, so I'll get that to you in just a second. All right, Colleen, off to you.
All right. Good afternoon, everybody. I think with all the time zones, we're definitely either afternoon or evening for everyone. Just a little agenda for today's webinar. So we're going to do a little introduction on myself. Going to do a little bit of background on soil and talk about the sensors that we can use to investigate soil. And we're going to kind of go over experiments and best practices at the same time, because I find it's easier to talk about them together because that's how they nicely go together. So a little bit about me. Hi, I'm Colleen. I have been at Vernier now for just over 10 years. It was a biology major and originally went into research where I was looking at human genetics and decided I did not like talking to fruit flies. I like talking to people a lot more communication taking place there. And so I actually became an alternative certified teacher and taught in Houston, Texas for 6 years, where I taught biology, I taught pre-AP biology, and I taught AP environmental science. While teaching, I got my master's in science education from Montana State University, which was an amazing master's program. I recommend it to everyone. And other things I did when I was teaching, I taught AP Environmental for 5 of the 6 years and was an AP grader for 3 of those years, which was oddly one of the most fun things I have ever done as a teacher. I really, really enjoy it. So let's dive into some soil now. So when I taught AP Environmental Science, soil was always kind of one of those tricky topics to kind of do labs with, because there's kind of a mixture out there of what you can do, and it's one of those that it literally is playing with dirt. You're gonna get dirty. So it can be a lot of fun to actually play with because of that dirty factor with it. Now, what's interesting is with soil, you have to remember it's this mixture of organic and inorganic matter that are going to be mixed together. And what's kind of cool to think about this is that really in soil, you're going to have essentially 3 different states of matter. You have solids, you have liquids, as in water, and then you're going to have gases and CO2, oxygen, and just atmospheric gases that will permeate through the soil as well. So it's kind of a cool, weird thing to look at. And it was something I always loved getting a sample of soil for students to actually look at under the microscope, because it kind of gives them an idea of what actually all the components in soil are. The other thing to remember with soil is that it is a mixture of a solid along with a whole bunch of pore space. Typically, we talk about it being about 50-50, where it's 50% soil, 50% pores. Some statistics put it at 55% matter, 45% pores. I've seen some variations on those values. And it really kind of depends on the type of soil we're talking about. And when we talk about the pores, they're actually a very critical component to soil and the health of that soil, because making sure you have these pores between soil particles is, one, how gases can move through the soil, so oxygen and CO2, and it's also how water can move through that soil as well. So it kind of ties into very tightly some of the specific soil characteristics that we tend to look at when we do soil studies. So some of the various characteristics are going to be things like porosity. So basically, how big are those pore spaces and how many pore space and how many are there? This is going to really impact things such as infiltration, like how well water can actually infiltrate through the soil. Then you have bulk density, which is kind of related to porosity because it's also looking at essentially how densely packed that soil is. So kind of in this little graphic here, you're looking at how big those spaces are. So essentially, how much volume is that mass of soil actually taking up once you remove all of the water from it? We also tend to look at things such as the structure of the soil. There's different structural components of how the soil sticks and molds together. Color. There are actual, if you have never looked at a soil color book, it is fascinating because of all the different types and colors of soil that are out there that are greatly influenced by the various minerals and nutrients and organic matter that can be found in them. We have soil texture that we often look at, soil pH, or in general, soil chemistry, and then soil temperature as well. So these are all different characteristics that usually when we do some soil investigation, we're going to be looking at. So one of the big things that we have to make sure when we talk about soil is looking exactly at the texture of the soil. And that texture of the soil is actually going to determine what type of soil we're actually looking at and dealing with. Now, texture is going to be dependent on 3 particles that actually make up the actual soil, and that is going to be sand, silt, and clay. And it's kind of one of these baffling things to talk about with students because everyone always thinks of grains of sand as being very small. But when you look at them under a microscope, they're actually quite large compared to the other components of that make up soil here. So we can look at, I love this diagram that I found. I've seen a whole bunch of different versions of this where you kind of see this nesting diagram of a picture of a grain of sand and how that compares to a grain of silt and a grain of clay, to really give you an idea of how different the sizes of these different particles are. And these particles are going to have a direct influence on a lot of those soil characteristics, such as porosity and bulk density. It also is going to have a big influence on what different types of plants can grow in these different types of soils, and some of the characteristics of how they can hold on to minerals and things like that as well. So to help us identify our type of soil we have is you use that lovely soil triangle. If you have not seen the soil triangle, there are soil triangles, all sorts of ones out there. And there are essentially two tests you can do to help classify your soil. And the simplest one that I like to do a lot was the jar test, where you literally just put a sample of soil in a jar at a known volume of water, add a little bit of different ways I've seen you can add a little bit of soap to it or a little bit of alum, and you shake it up and then let it settle for 24 hours. And as it settles, it's going to be, because of those particle size differences and the different densities they have, is that sand will settle all the way to the bottom, and then you'll have a layer of silt and a layer of clay. And so you determine how thick each of those layers are, and that's going to give you your percentages that you can then use the triangle to figure out what type of soil you have. The ribbon test is a little bit more, I would say, objective than subjective. Is you're actually going to be adding to you're going to be adding water to a sample of soil and creating kind of a ball, and then mushing that ball out to create a ribbon to actually see how long that ribbon goes. And then based on that length of that ribbon and the texture of the soil helps classify what type of soil you're looking at. On the slide here, you can see I have a link. So when you skip this slide deck, you can click on that link, and that actually takes you to a great website that has both procedures written out. So in case you want to do that with your students. So I see there's a question about what does the alum or soap do to the soil? So that's a great question, Emmy Lou. When I was doing it, I was told the reason you add it is that it helps actually separate those clay and silt particles more, because they're such fine particles that you want that little bit of separation to take place. And it just kind of helps further do that. I don't believe, if I remember correctly, it was usually just like a pinch or a dab. It wasn't like a large volume of it. And one of the ways that I was able to do this in class with my students and have a lot of fun with it is, in my AP class, I actually kind of turned it into a project learning situation where I turned us into soil scientists, where teachers, I put out a call to teachers in the school to bring us soil samples from their gardens, and that the students would then identify and go through and look at these different characteristics and identify what type of soil the teacher had and the different characteristics of it, and give them recommendations on how to improve or change the quality of their soil. So it kind of created a fun thing of having them turn into the scientists and do actual soil science with these samples. So one of the big things of why I talk about texture so much is texture really influences a lot of the characteristics in soil. So I found this great diagram here that kind of shows the differences with sand, silt, and clay. And you can see kind of the size of the particles here. And with sand, it's got great size of pores. It's these huge pores, which is nice, but that also means that water is going to drain through really, really quickly. But it also has a lot of room for roots to be able to grow and move through that matrix there. In more silt-heavy sand, you still have some good draining that takes place. It doesn't drain as fast, and you still have some good movement for actual roots to actually move their way through. Great and easy for organisms to go through. And then in clay, it's kind of the absolute other extreme from sand, is that it really, the water cannot flow through it. Air can't flow through very well either. So it's going to be really, really easy to compact. Nutrients are going to settle in, salts are going to settle in, and roots are going to have a really hard time moving through it. So it's kind of one of those, it's kind of this Goldilocks syndrome where you want a nice mixture of all three components to get this kind of perfect soil, because they have all important characteristics. Okay, so what sensors can we do for soil, studying soil? So some of the sensors that I used with my students was looking at soil temperature, moisture, pH, using a conductivity sensor to look at salinity. We can use carbon dioxide sensor, which I'll do last because that's a very fascinating thing. And I'm just going to talk a little bit about how we could use a turbidity sensor as well. Um, the turbidity sensor is more to look at, you can create a situation where you study the effects of runoff on soil and how you can have basically different shoe boxes that have soil in them, and then you can plant different plants or have different substrate and pour water down it and then collect the water sample. And you can show how if you have, for example, a bunch of grass, how not much soil is going to wash away because the grass is really holding on to it. While if you have a more barren area, the soil is going to wash right off. And so you can use a turbidity sensor to get a quantifiable value to show how much soil is ending up actually in your water sample. So the other thing I want to point out here is with pH, we have two different types of pH sensors that you can use. There is a flat pH sensor and our regular pH sensor. The big difference here is whether there's an exposed glass bulb or just a flat junction. Both will work great for soil. I tend to recommend the flat pH more often than the regular pH, just because the chances of a glass bulb breaking on a little piece of rock or something like that are going to be a lot higher than being able to break this junction here. So that's kind of, you can get by with either one of them, but I tend to recommend the flat over the regular pH. All right, so let's talk about measuring soil temperature. So the big thing with soil temperature that you have to remember is that soil temperature is going to vary on depth. So you want to make sure whenever you're measuring soil temperature that you're measuring it at the exact same depth in every place. That way you can easily compare them together. You can also set up a really interesting experiment in the classroom to kind of demonstrate how solar heating can affect the different types of soils and how deep it goes. Is you basically have a water jug that you cut off the top, and then you have 3 sensors put in at different depths and put a light on it. And then as that runs, you can see how the temperature changes between these various factors. If you are going to be just doing temperature out in the field, what you would want to do, I'm going to switch my sharing screen now. You can see all of my toys over here. What you would want to do with the temperature probe is actually make a measurement of how deep you want the probe to go into the soil. And then you will want to take a piece of tape and wrap that tape around that point. That way, you know that when I insert my temperature probe into my soil, I'm going to go up into that tape point. That way, it kind of ensures that you're doing the exact same depth and you're not getting this variation that you could based on different locations where you're reading. The other thing to note is with a temperature probe, the active measuring point is going to be the very tip of the sensor. So that tip is where you're going to actually be getting that temperature from. The other thing to keep in mind is this is a stainless steel temperature probe, so it's going to take a little bit longer for the temperature to kind of settle in. So you can't just shove it in, put it in the soil and immediately take a temperature. You're going to want to let it sit for a minute or two and wait for the value to kind of reach an equilibrium and then keep that value there. Right. Now, soil moisture. So Basically, when we talk about measuring soil moisture, we're looking exactly as how much water is present in that pore spaces in the soil. And so if we think about our pore spaces, remember earlier I said that it's usually around 50% solids and 50% pore. So when we have a soil moisture reading, realistically, that soil moisture can essentially never be higher than 50%. So your soil moisture readings are always going to be below 50%. And if they are close to 50%, you're not so much going to have soil as much as you're going to have just muck or swamp. So typically it's going to vary based on our type of soil of what our soil moisture readings are actually going to be. What's really cool with the soil moisture is with a sensor, you can actually test permeability of soil by sticking the soil into a sample of soil and then pouring water through it and seeing how quickly that water can reach that sensor and how quickly it moves through. One of the key uses with soil moisture is that you want to use it horizontally, not vertically. And the reason is if we look at a profile of soil, soil has lots of variations as it goes down vertically here. So you want to make sure if you stick the sensor straight down, you're going to be getting measurements through all of these different layers that are going to have lots of variation in them. So what you want is to stick the sensor in through the side to actually get a uniform reading of the soil in that level there. And that means you're going to want to dig a trench and then stick the soil sensor in there. Or if you're using a container, which I'm about to show, you want to stick it into the side of the container to get a reading. So let's do sharing here. All right, so you should see now graphical analysis along with my table of toys. So I am going to turn on my soil moisture sensor. Sensor data collection. Connect. And with my graphical analysis here, I'm going to just turn on my meter mode instead of looking at time-based data, because I just am going to do a point-based reading of what is my soil moisture. So you can see it's reading 0 now because it is in the air. One way I can test my soil moisture is just putting my hand over it. And you can see that the readings are going to change because it's measuring essentially conductivity that's taking place. So I have a Tupperware container here that I went outside and filled with soil. And in this Tupperware container, I've cut a small slit. There we go. I've cut a small slit here so that I can actually insert my sensor in that location. So I'm just going to slide my sensor in. Give my soil a nice little compaction. And I can see that I'm reading at around 16.3%. So that's about how much water is going to be present in that volume of soil. Now,
Colleen, we just got an excellent question.
Oh.
Does the soil moisture sensor need to be calibrated?
No, it does not. We have the soil moisture sensor comes pre-calibrated. We calibrate it here and it is ready to go as soon as you plug it in. Excellent question.
And while I have you distracted, that's just the regular old conductivity sensor you're using, right?
No, this is a specific soil moisture sensor. I am going to be doing in the next slide or two using the actual conductivity probe. But essentially the sensor is using it. The engineers can describe it a lot better than I can of exactly how all the electronics work here on this. But it's essentially looking at water potential and current coming through and detecting that to give an idea of how much soil moisture is present. Any other questions while I was going? And for you guys that have not noticed, Melissa Hill has joined us. She came in a little late. She is our one of our staff scientist chemist and director of chemistry. And she is here to help me out and answer questions or let me know when questions come in. Oh my goodness. Let's see here. Oh, other good questions. When doing a composting experiment, would the soil moisture work in the substrate? Yes, so you could, it could be used for this type of experiment too, is that you could use it with your compost to get an idea of the moisture content and level of compost that you have in there. Yes, so I did put the sensor in sideways instead of flat. One of the reasons I put it in sideways is because I want to make sure I kind of get a nice zone of reading around the sensor itself. Flat will work, but I tend to like putting it in this orientation instead, because it's one, easier to insert that way into my different soil samples. And usually by going in this way, you have the entire plane within a band of a specific type of soil in there. Other questions? Okay, the other thing I like to point out is once I remove my sensor from the soil, I now am going to have a hole there where that sensor was. So, oops. So you're going to want to have either dump the soil out and put it back in, or get a new soil sample to do your measurements. When you're in the field, basically, you'll just want to do some measurements at different locations instead of all in the same spot. All right. How long can you collect data before it shuts off? That's a really good question. You basically get to decide how long you want to collect data for. Right now, these are set that they can collect data for quite a long period of time. But right now you have to be connected to an active device such as an iPad or a computer while it's doing its data collection. All right, so back to my presentation here. Yes, so strength and durability. I do a lot of testing of these sensors to make sure that they are very sturdy. The original design of the sensor I rejected because I was able to break it in 5 minutes by bending it back and forth. So this one is very robust. I basically would say if you aren't able to get it inserted in, it's probably not going to be a good way to use that sensor. So if it's like super, super rocky soil, it's gonna not go in as well and give as good of a reading. And that's kind of where in my slide here, you really want to make sure you're in pure soil. And if it is that dry that it can't actually get inserted in, then I would not recommend using it in that situation. On the same hand, you want to make sure too that you're using a very uniform compaction, because it kind of goes in with the idea of the porosity and bulk density, that the more you press down and compact it, the less pore space you're going to be having. And so you're actually going to be kind of moving water in to less pore space, and it's going to give you some very variable readings. So you want to make sure you're compacting to a point that is essentially equal to the soil that you're actually sampling. Yeah, so I do see if you, so Kathy has a very good question on, does the sample change depending on if you dig a trench or add to a container? Yes, so for this sample, I wanted to bring a container in so I could be able to demonstrate it. I typically would prefer to do it in the field. That way you have a more uniform area that you're measuring. For me, I had to dig down to get soil, and so I ended up mixing up a bunch of layers. So the actual moisture reading might not be super accurate compared to what it is outside. But if you're doing something in, like, for example, I did bottle biology tubes with my students, and would have just slits cut in the side for them to put soil moisture sensors in on that side. And usually what I would tell them to do to kind of help close up the hole that they created is kind of wiggle the sensor back and forth to kind of help the soil kind of fill in those gaps as they went. And they were only measuring them once a week, so that kind of allowed the soil to settle and kind of compact on its own there. And yeah, Diane, very heavy, dry clay would also be very, very difficult to get good soil moisture readings on. All right, so moving on, let's talk a little bit about soil chemistry. So with soil chemistry here, let me make sure everyone's still seeing my screen. Okay, good. With soil chemistry, the thing we have to remember here is that with pH probes and conductivity probes, is they're really made to be used in aqueous solutions. So you want to use them in liquids. So you can't just stick the sensor directly in the soil and hope to get a reading. One, that can actually break your sensor, especially when we're talking about pH probes. And two, it's just not going to give an accurate reading for that. So the first thing you want to do is actually take a soil sample and create a slurry. So you're essentially going to take 1 part soil, and you want to add either 1 part water or 2 parts water. I've seen variations on this. In some places it says 1, some it says 1.5, some it says 2. It just kind of is going to vary on what you feel like. The other key point is that you want to make sure you use distilled water, because essentially what we're doing is taking the ions that are essentially bonded to the soil in there and pulling them out so that we can actually measure them in the liquid. And once you have your slurry, you want to shake it up and shake for a little while to make sure all the particles get broken up and have contact with water. And then you're going to want to let it sit. And I've read anywhere that usually what I would do is I would let it sit overnight and have the students measure it the next day. Some people say just 15 minutes is fine. So you kind of have this range of at least 15 minutes or even longer. And before doing this webinar, went out and go. Just the button again. There we go. Went out and collected some more soil, and you can see I created my own little soil slurry. So I added about, it's trying to do this backwards here. There we go. I got about essentially 100 ml volume of soil and added that to my Nalgene bottle here. And then I added around 200 milliliters. No, I had 100. No, I did add 200 milliliters of water to it. And then I shook it all up, and you can see it separated out quite nicely there. And while it's mucky, this top part is all water. So what I can do now is actually stick my sensor in this top portion and actually get a reading. So to do that, we're going to connect. First, I'm going to do pH. And I'm going to use a regular pH sensor because it's what I have readily available. And this is a pH sensor that has actually been sitting in a pH 7 buffer, which is not what you should normally be storing it in. It should be stored specifically in a pH storage solution that is a pH 4 buffer with potassium chloride added to it. This, though, I tend to, before I use them, I will actually soak in pH 7 because it kind of brings it up to more that neutral pH, and it allows the reading to kind of stabilize and happen quicker for me, because usually our pHs are going to be closer to around the neutral range. So I'm going to take my pH out of the solution, rinse it off. Keep looking over my shoulder, make sure people can see what I'm doing. I'll rinse off. And then I'm going to stick in my water slurry sample. And I want to make sure I'm just measuring in the water. I don't want to go down into the soil. So stick in here and kind of see where we end up. Now, being that we are in Oregon, our pH of soils tends to be a little bit more acidic because of the number of conifers we have around. So I'm not too surprised that our pH is dropping here. And that's kind of around where I would expect our pH to be, is between 5 and 6. So that's looking pretty good. And you'll see what I'm doing is I'm not taking a reading right away. I'm kind of watching and monitoring. And essentially, we want to make sure that our pH kind of reaches a stable point where it's not seeing direct movement down or direct movement up. It's kind of now going back and forth between 5.78, 5.79, 5.8. So this to me is right where my pH is going to be reading and what I would record. And then once I'm done doing that reading, I want to rinse my pH probe off. And since I used one with a glass bulb, I'm going to make sure to rinse around the bulb to make sure I don't have any particles around that. And then go back into my storage solution. Because one big thing with pH probes is we want to make sure that we don't leave them dry. They are not happy when they are dry. And I'm going to disconnect my pH. And now I'm going to do a conductivity sensor. And we can see what the conductivity of our soil is. Again, we're going to go to meters, turn off our graph. And same thing, I'm just going to stick my sensor in. And we have a conductivity of 25 microsiemens per centimeter. So fairly low conductivity. And I will talk about that in a second of what that means. Okay, so questions that have popped up since I've been talking a lot. Looks like Melissa answered the lab quest by Vernier. So yes, so you don't want to put the sensor directly in the soil because one, you're going to break the bulb. It's a good potential to actually break the sensor. And two, it's not going to really give you an accurate pH because you really need to have a solution in order to get an accurate pH reading with it. So that's why we create those slurries. So back to the presentation here. All right, so why we're measuring these two factors here is one, pH is actually very important because the pH of soil greatly impacts the availability of nutrients and how easily plants can uptake those nutrients. And so we want to make sure the pH is in the accurate range for the nutrients we want our plants to take up, or for other factors as well. For example, if any gardeners out there know with hydrangeas, you can change the pH of your soil, and that actually changes the color of the hydrangea flower you get as well. So it can be a very important measurement. Conductivity is a way for us to look at essentially the level of salinity of the soil. So we're really looking at how many ions are actually present in the soil, essentially salts. And this is important for when we think about agriculture and irrigation, that depending on different locations and different types of soil, that water might have a lot of minerals in it. And if you keep irrigating and it keeps evaporating away, it's going to leave a lot of those minerals behind, which can actually increase the salinity of the soil. And over time, it can actually make the soil not be as functional anymore and not work as well. So it's very important. Yes, and so actually the pH probe and conductivity probe are what I would say more commonly associated with being used for water quality, not soil quality. So it's kind of nice to show that you can do, you can measure not only water with these, but you can do soil measurements with them as well. Okay, let's moving on. So one of the more abstract things to do with soil that I really enjoy and love doing is looking at soil respiration. So a lot of times with students, they don't think of soil as being a living thing. They just think of it as dirt and that's it, and not realize that there's actual organisms inside the soil growing and living. So a really cool way to measure this is actually doing what we call soil respiration, where we can use a CO2 gas sensor and put a bottle over an area of soil and actually measure how much CO2 is being released by essentially microorganisms and other macroorganisms that are present within that soil. The key thing that I wanted to try to show in these pictures I took here is you want bare soil. So if you're doing this in like a garden that has mulch on it, for example, you want to make sure to clear the mulch away and get the respiration chamber directly on the soil itself. And this is the same for if you're taking soil samples to do slurries, to do the chemistry measurements. You want to make sure that you remove that top layer of what we often call duft, and you want just the actual soil. You don't want that organic matter that's just sitting on top. Any size chamber works. This is an old water sampling chamber that I took that I cut apart. And I know the exact volume of it. I know it's about 500, I know it's 500 milliliters. And we sell these like grommets here that will actually allow you to create a hole and then put the grommet in for a CO2 sensor to fit very nicely within that chamber. If you aren't able to go out and do this experiment and look out in the field, you can just get a scoop of soil, bring it back to the classroom, and actually put the soil in the Nalgene bottle that the CO2 sensor comes with, and just measure the respiration of the soil that way as well. One really interesting thing to do with this is looking at the effects of different types of soil of compaction has on soil respiration. One big problem with soil is as it gets compacted, it basically lessens the amount of pores available and reduces the amount of gases that are available for organisms to do cell respiration. And so I did an experiment around here where we had some soil that was highly compacted and I saw basically no respiration activity. And then I went to another area that was a bioswale, an area that helps absorb and dispose of rainwater and surface water. And the respiration rate was through the roof on that one, but then went to a garden box that actually had compost added to it and saw even more respiration because of how active that compost was with the organisms in there.
Colleen, we got a great question asking about how long in this application you're talking about, how long would you want to collect the CO2 data for?
So you can collect, I usually, when I do it, collect only for about 5 minutes, but you can go up to 10 or 15 if you would like as well. Usually I try to make sure my experiments don't go too long because biology experiments tend to be a lot of hurry up and wait. So I try to find my minimum, which usually is around 5 minutes to be able to do this experiment. And I have had great results with it. I do it out in the field a lot where there's a workshop that I do out in the wilderness, and we look at soil respiration rate of trail versus about 3 feet off the trail versus 10 feet off the trail. And you can really see the impact that humans have on soil compaction and the effect that has on soil respiration rate. Any other questions?
There's been so many good questions with this group.
Very good
Loving
question.
it.
Yeah. And so the big thing with soil is have fun with it. Like find different ways to get students really interested in it and can really get into it. Like people joke with me here in the office because I've been doing so much soil moisture testing that I'm literally getting paid to make mud pies right now. And it is a lot of fun to see how different things can come together to be able to help us better understand actual soil science. Against the soil, does the soil respiration sensor sit against the soil inside the bottle? So in the picture here, I try to have I've only put it down a couple centimeters so that way it has a good stable hold so it doesn't fall over. And you don't want the sensor to directly touch the soil because you don't want actual dirt particles getting into the sensor. But you want to make sure you have a chamber for the gases to kind of build up inside as well. Hopefully that answers that question. All right. Well, thank you all for coming today. I'm not sure if there's any other questions. Feel free to put them in chat if you have any. But thank you all for joining us, and hopefully this will inspire you to get outside or bring some dirt inside and have your students kind of play and get used to what dirt and soil is.
And I wanted to mention before everyone goes, I forgot to remind you all at the beginning, but we actually are doing a giveaway. So everyone who attended today was entered to win a Go Direct pH probe. And I've run a random name selector, and today's winner is Greg S. So congratulations, Greg. I'm going to shoot you an email here in just a moment. So keep an eye on your inbox. It'll come from an @Vernier email address. So congratulations, and please give us your feedback in the survey link I just dropped in the chat.
All right.
All right.
Yeah, thank you all for coming. And if you have more questions, don't hesitate to reach out to biology@vernier.com. You will definitely reach me through that process. So everyone have a wonderful evening, and thank you again for joining us tonight.